Let's wait 2 more minutes. Can you hear me well? I don't need to speak in the speaker, no? Okay. We are missing people, no? Okay. Okay, hello. I'm Sebastian. I think I know most of you, except for a few. Okay, so what we're going to talk, I'm going to be, I'm one of the persons organizing this class together with Mona, and I will be here in many of the classes later on with the guests, but I will formally give this, I think it's only these 2 classes, today and next week. And the idea is that today I'm going to give you some descriptive basic ground that will help you guys understand more or less what is the neurochemistry of a neurodegenerative disease. It is very general, but I think it's really very useful to know some of the. You guys can maybe to speed up or to go slower depending on how you feel with the stuff. Okay, so feel free to interrupt me, to ask any questions you want, or to tell me this was not clear, go over again. Just, I mean, the pace we will go depends a lot, much more on you than on me, and we will go in the lecture as far as we can. Okay? So, we always have this view that our body is a beautiful and perfect machinery. And I heard so many times when I was studying biology, and especially from my friends who at that time studied medicine, how astonishing is the body, how how perfect design, how everything is so equilibrated and so perfect. Okay. However, if you learn a little biology and more, if you know a little physics and a little thermodynamics, and if you study a little immunology, for example, you would realize that indeed the miracle is not how the body works. The miracle is that the body can keep working. It's a miracle that we are alive, because we are all the time in the verge of collapse. So I see, instead of seeing the body or the human body or the human biology like this, I like to see that we are a machine on the verge of malfunction. We are in equilibrium like this. And many things can tilt us to be like this and to have disease. So at the end, anyway, always collapse. So a perfect machine works forever, and our body doesn't. So, and this is because the cells really operate in a quite narrow window of conditions. And if you think in a stone is going to be a stone in a huge range of temperature, but a cell will be a cell only in the very narrow range of temperatures. I would say no more than, if you take the most extreme environment and conditions, no more than 100 degrees range. So if you change the temperature, you're going to decrease the enzymatic activity. You will have the proteins which are going to be doing what shouldn't be doing, or not doing what should be doing. And also, a lot of the processes in the cells are mediated by diffusion, or by facilitate diffusion. So this diffusion will really be much lower if you decrease the temperature. So really the cells operate in a quite narrow range of temperatures. This is really not completely relevant for humans, because our body anyway keeps 37 degrees. But other factors start to be more important, like the ion concentrations can be critical for a cell to survive longer or less, even to explode, to be poisoned, to lower the enzymatic activity. Many ions are used as signaling molecules, so the presence of these ions can be interpreted by the cell as a signal and be very stressed over the cell. Also the nutrients, obviously, and many other things. So our body is subjected to changes in conditions every day, both from the outside and the inside. And it is very important to have systems that minimize these changes, at least at the level of the cell, the single cell. Okay? So this comes the concept of homeostasis, and I believe most of you may know what homeostasis is, but this is I would say the proper definition. So homeostasis, which is from the Greek homeos, and which means similar, and standing still, is a property of a system, either open or closed, that regulates the internal environment and tends to maintain a stable, constant condition. So you want system that will keep you maintain a stable condition, and it's typically used to refer, so you guys know how to read, but we shall say it anyway, typically used to refer to a living organism. The concept came from of a milieu interior that was created by Claude Bernard, and okay. So the idea here is that multiple processes which are in dynamic equilibrium make homeostasis possible. So basically, if we see from the outside, we can have a wide range of many things. You can start thinking temperature. Okay, we are in winter, we are in summer. In the environment, the conditions in the environment change all the time. And we have a system that makes that the conditions that reach the cell are really, really much more limited. And the failure of the organism to filter all this variation, or to let's say to really accommodate these parameters in a very narrow condition, is what leads to disease and death. So how is our body maintain homeostasis? I'm going to try to go more or less quick over this. One way is by negative feedback loop. You know, you have a process, a factor, which when this factor increases, there is a sensor that can detect it and can somehow inhibit this factor. So, you guys know an example of this type of thing, physiological feedback loop?
Many.
Eh?
Many. Like in the HPA axis.
Yeah, for the hormones, yeah, exactly. For the heart rate, this is also, you know, the body uses this feedback loop system all the time to maintain homeostasis. Okay. Okay. So what they wanted to remember until now is that normal cells have a very narrow range of function or steady state, which is homeostasis. You can even think, and there's people who have you know, whole books about this, that the only reason, or the real adaptive value of us to have a body is to be able to maintain our stem cells in homeostasis. Okay? Because if you have a unicellular organism, you know, the unicellular organism in the seed is much more fragile to change in the environmental condition. But if you have a whole system that will protect this a few number of cells, which allow the organism to reproduce, this will explain why we have a brain, why we have a body, you know, why we are. Okay, that's the philosophy. But I think I try to bring this concept. So, if we have excess physiologic or pathological stress, this may force the cell to a new steady state. And you may go to a steady state A to a steady state B, and we call this adaptation. And not always adaptation is good, because we are going to talk about this. Many the situations in which we have in cells in which they will later be neurodegeneration is because there is adaptation to chronic stress. And this adaptation to chronic stress, in the long way, will lead to the death of the cell. Homeostasis is not adaptation. Homeostasis is system that will avoid the cell to suffer oxidative stress. So will be all the mechanisms which will prevent the cell from having oxidative stress. Okay? Okay. Too much stress, when too much stress exceeds the data of this capacity, we have injury and death. So I'm going to go quick over this because I want to focus on the part, but basically the synapses are very sensitive to stressors. So there are structures which are very, very complex and very, very sensitive to stressors. One of the, especially the ionic stress, oxidative stress, okay? So, and it is known that the age conspires against the capacity of the neuron to keep homeostasis. So, when we talk about neurodegenerative disease, we talk about age. But why you guys believe we talk about age? What is the problem with the age? Okay, this is a fact. Yeah, good. You are older, you have more chance to get. But why you have more chance to get?
Well, the theory in the field was that as you age, you accumulate damage. But now, actually, they're talking about a different theory that your defenses collapse and then.
And you work on it. You work in aging, no?
I work in aging, not in the brain, but.
Yeah, okay. You gave us the right answer, but you know the common sense answer will be you have cumulative damage over a lifetime, and you have cumulative damage over the lifetime. All your stress exceeds some threshold, and you have death. But there is many facts that suggest this is not the case. But today, what I'm going to talk is mainly this old theory. Okay, mainly we are going to talk about cumulative damage in proteins, in membrane, in DNA, okay? But for you guys to know, now there is many facts that suggest that it's not the cumulative damage, but there is a program which is designed to make you more susceptible and eventually to make you die, okay? And I think Wood Cohen will be talking about this, so I will try to convince you that this is what happened, so he come and tell you that this is not what happened, okay? But that's more or less the view in the field. It's not clear one way or the other, okay? And probably both of them together. So in age, you have oxidative stress, metabolic and ionic stresses, and you have two systems. Basically, you have the stress, and you have the system which will prevent you to get the stress, and the system which will repair the stress. So if you repair the stress, you will have a healthy individual. But if the damage is more than the repair, you will have accumulation of damage, protein, DNA, and membranes. You will have neurodegeneration. So basically, here we talk about an equilibrium between damage and repair. And this equilibrium, usually during a young age, is okay. Most of the stuff is repaired. But a little that is not repaired accumulates over time at the rate and at the level in which, when you are old, you are not going to be able to repair as much as you damage, and you get to neurodegeneration. I want to say that I think I have it afterwards, but why we talk about neurodegeneration and not the leg degeneration? You know, what is different about the neuron than about the leg? Of course, all the body, the degenerated by the neurons are much more sensitive and don't divide at the same rate as other parts of the body. But obviously, the aging process impacts every single organ and everything. Okay. So, age is not the only factor. Of course, we can consider a key factor. And you have many environmental things like exposure to stress, for example, or high calorie intake, physical and mental inactivity, toxins, iron, infectious agents. So you can have many environmental factors that can, I would say, like advance the onset of the disease. And you also have many genetic factors that can help you develop the disease. Okay. So in most cases, I think Mona talked about this when she talked about Alzheimer's and Parkinson. I mean, most of the cases of Alzheimer's and Parkinson seem to be sporadic. They don't have a clear genetic component. And this is, this is what we know until now, seems to be the case. But a small proportion of the cases of Alzheimer's and Parkinson and many of the other neurodegenerative diseases a small portion do have a genetic component, a strong genetic component. And that's in some of these mutations that we put, although like the alpha-synuclein polymorphism is very famous, and the apolipoprotein, and the APP and penicillin mutations. Yeah.
Outside the young mutations, if we take, for example, a twin study, if one twin has Alzheimer's, what are the odds for the other one?
Yeah, we are going to talk about this. So yeah, there is of course there is a lot of subtle accumulation of mutations that can't help explain. So there is an important component of these diseases which are genetic, but they are not monolithic, and they are not like when you have these mutations.
Okay, but we have now traits like height, which we know is 80% genetic, but we cannot point to G.
I'm going to talk about the genetic study. We're going to talk about this. And also you have some mutations that help you not to develop these diseases. Some polymorphism in ApoE2. I don't know if we want to talk about this. And also it is known, basically, if you if you live like a monk, then you may or if you smoke, you may have less chances to develop some of these diseases. So the two key questions here in the neurodegenerative diseases, and I should say that there are many talking here about Parkinson and Alzheimer's. Okay. So if the cellular damage is progressive and cumulative, and the question is why? And neurons are more susceptible to these stresses, and they are not replaced. Okay. There are cases, I guess most of you may know some of these studies when they show some of the, at least some part of the neurons can reproduce, and they are very clear, it is true, but it is not a common case, and it is not happening all the time. Okay. So while they are more susceptible, so let's just go a little further and think what are the key characteristics of neurons. First, they don't divide, so the divide is cumulative. They use a lot of energy, and this is very important, that most of the glucose we take goes to the brain. So if using the energy in this aerobic way leads to a generation of free radicals, which are very, very toxic because they generate a lot of oxidative stress, which usually the body can, is fine, speaking, everyone understand me? Yeah, fine? Not you guys in the back? Fine? Yeah. So this is the thing, and most of them generate toxic compounds. And this is also something that sometimes is understated. So many of the neurotransmitters, like dopamine, really the pathways to make them are very, very toxic for the cell. As a subproduct of this, the generation of these neurotransmitters, there is a lot of substance that can produce oxidative stress. Okay. Another thing that shouldn't forget, yes.
At least in nematodes, as far as I know, they show that when you increase oxidative stress by deleting the salts, for example, you don't get shorter lifespan. So this was the free radical theory. It was very popular for many years, but now people question it. There was showed evidence, for example, in the ear, in mice models where oxidative stress is known to affect lifespan. But in the brain in general, I don't think anyone...
I think you even insult. That's what I recall at least. If you have a model, at least in slides, if you have models of a disease, and you delete salt.
No, I'm talking about the normal condition.
Yeah, normal condition maybe not. But you may need the salt. I see what you mean, because you say maybe not for the, not general homeostasis, but as an adaptation to...
They have higher levels of oxidative stress, but they have a normal lifespan, as long as they don't eat food.
Yeah, that's the thing. Okay, we can... But he's giving a good point. If you are not under stress, you may not need some of these defense mechanisms. I think what it shows also is that who knows what we are every day. Probably we are under stress, drinking in a plastic cup. I mean, I'm not talking about Parkinson in general. We may get the stressful conditions which make the thought, but I get your point. It is, that's what I said, this is the canonical view, and many of these things, even when I go to talk about aggregation, many of these things, if you ask me what I think, I don't think it's like this, but I think we need to know the common ground. What is the ground in the field for the last few years, and just in order to destroy it. Okay, and the cells have very big surface, and this is, especially in mammalian cells, the surface of the neurons huge. So, in this, you could argue that the total exposure of a cell to stress is more. Why do neurons die in neurodegenerative diseases? I'm going to give the classical view here. One possibility is protein misfolding, oxidative stress, or apoptosis. And let's go to look into each one of these possibilities. Okay? So this is a cell, although it's not a neuron, okay, but it's a cell. Everyone knows. I think most of you guys are biologists, not here. Yeah. So others, yeah. Okay. So you have a mitochondria, you have a lysosome, and you have a ER. And I think I was trying to point out of the places in which you could have oxidative stress. So the genetic information, all of you know that it translates and the proteins are fold in order to get the protein. So a huge process in biology, which is not understand, I would say almost at all, is how a linear amino acid change can fall in a reasonable amount of time into a unique functional structure. And when this happens, it's a very complex process which happens very quick. And you have a lot of folding intermediates and hydrophobic domains. And during this process of folding, things can aggregate because hydrophobic residues from inside the protein can be exposed and interact with each other instead of interacting with themselves. So folding is a quite important process. And when you don't have proper folding, many of the times you don't have proper folding, you have neurodegeneration, at least in some of the models, like in flies, and probably, I don't remember in the chapter on the division in the worms, also gives rise to neuronal death. Yeah?
Can you just connect the dots for me? Why do you have neurodegeneration when you're protein?
I think this is what we're going to see later. No, it is here. You mean this? We are starting here. I just tell you that the protein needs to fold in order to work. And when the protein doesn't fold, you may have aggregates because the parts, usually the internal parts of protein are hydrophobic. So instead of interacting with themselves to close themselves, they interact with each other. You may have now an aggregate. Aggregate of many different proteins. Okay. So there is many processes that try to assure that the formation of aggregates in the cells doesn't happen. One is the co-translation quality control. I don't know if you guys heard about this. No?
And CS is known, most of them don't even mature.
So when you have a nascent peptide, the protein is translated from the ribosome. Okay, you have a nascent peptide. And basically, what you want is the protein to fold with itself. Okay, so you want the protein to fold with itself. So you want to favor the intramolecular interaction, and you want to try to prohibit the intermolecular interaction. And I would say that many times because you have different hydrophobic domains in the protein, they will be maybe far apart in the sequence. So the intermolecular interaction may be much more likely to occur at this stage. So there is a system which is commanded mainly by two chaperones, HSP70, HSP40, which binds to the nascent proteins and prevent the intermolecular aggregation. Keep them open until the whole domain is synthesized. Okay? I don't want to go into the details into the system. I think you learned this in the advanced biochemistry. So this assures you that when you are making a protein, there is no intermolecular folding or help you. So of course, if you have a condition in which this system is impaired, in which the intermolecular folding would happen, you're going to have now aggregate. Okay. So let's say you have a native protein. When this protein is subjected to stress, the protein can lose like for example, you can think temperature, oxidative stress, you can even think some chemicals, some ions, okay? The protein may lose the conformation and be unfolded or misfolded, and this protein needs to be quickly degraded, okay? Or needs to be refolded. So there is a signal that is an ATP dependent, which is going to refold the protein into the native structure. Okay, this is what we call assisted folding. The second way to prevent the aggregate formation when you have a protein which is under stress is by prevention of the aggregate formation. Both systems are driven by chaperones. Okay, and the idea here why chaperones are so important in a neurodegenerative disease, because they help protein folding in normal stress conditions. You have a protein native, and the chaperone you have the protein unfolded, and the chaperone can take you to the native state. So if you have now a stress which is going backwards, the chaperone will help you to get back. And of course, people working in structure of protein know this for many years. But I think at the beginning, in the late 90s, when people start to do a screening for neurodegenerative disease, and they start to find when they express chaperones, when they overexpress chaperones, they could prevent many of the symptoms of neurodegenerative disease in animal models. So I think that this was quite was sort of surprise to link these two things in the 90s. Now it's obvious. Okay. Okay. This is what I was talking before, the co-transcriptional chaperones, okay? And then you have a chaperone which is ATP dependent, which is going to help the final, the protein to acquire the final shape. How, I will just comment this. How do you know how the chaperone can help you fold properly? Someone knows how? No?
It can bind to the glyphobic regions and block it from connecting to each other.
Well, so basically this is one mechanism that we'll do, but the concept is that the protein needs to try millions of forms to get the right one by chance. We will help only to try a few conformations, and usually this is achieved by bringing some of the parts together by retaining the hydrophobic parts. Okay, there is many different types of chaperones, but this is the concept. So many of the chaperones were first identified as the heat shock protein, and because they're highly expressed under stress conditions. And the common consequence of these stresses is the presence of a lot. Okay, look, please, because usually in this class, most of the students are computational, and if you guys, all of you feel this is too basic, just tell me, okay? This year maybe that is not the case. If you have seen many of the things before. So so basically we have one step of quality control is at the co-translational level. Then you can have a quality control like will be unfolding a response, proteins get unfolded, you have a refolding system. And on the other hand, those proteins which you cannot fix, you need to degrade. Okay? And this is the UPS and the lysosome. And basically, you guys know the oxidative system. So proteins which are going to be taken to degradation, they are tagged with a small molecule called ubiquitin. The ubiquitin is a small peptide which is in an ATP-dependent way put into this cascade. You have the E1 and E2 ligases. Everyone knows this. And basically, at the end, you're going to have whatever protein that is sensed as needs to be degraded is going to be polyubiquitinated and be degraded. And there was a theory, okay, I'm not going to, this the machinery called the proteasome, which end up destroying the protein. Okay, let's just skip. You know all this, okay. So it's just talking about the ubiquitin system. It's okay? Yeah. Okay, I'm talking about the ubiquitin system, and this system is very, very important in neurons because the synapse is such a dynamic structure. It's changing all the time. The channels are changing all the time. The receptors are changing all the time. So all the time, it's a structure that needs to be remodeling itself. Okay? And usually you have proteasomes associated very closely to the membrane of the neurons and etc. 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 I'm not going to go into the details. Just want to say, and proper protein degradation is key for synaptic homeostasis. So the UPS, the ubiquitin system, regulates the recycling and the release of synaptic vesicles at the presynaptic terminal. Okay. So when you lose the concept, when you lose control of degradation, the action potential now is not going to be many times the neuron is not going to be activated. So it is very clear when the UPS system is not right, the neuron is going to lose excitability, is going to change its properties. So all what I told you this is protein quality control under, let's say, normal conditions. So what happens under stress conditions? So one case is heat shock, which you would say doesn't really happen to a cell in our body, but it's a clear case of stress. When you have a heat stress, protein folding will be inhibiting, and the cell has many systems to control this huge shock to the cell. One is to stop immediately protein synthesis, because if you cannot fold protein or maintain the folding of the protein well, you don't want to make more proteins. And this signal goes to the protein synthesis. You inhibit synthesis of any mRNA, you inhibit the splicing, and you activate the synthesis of these enzymes, or these chaperones, heat shock proteins, which are going to be the mediators of the refolding. Yes?
Mechanism of stopping translation, no?
I don't think so. I mean, the splicing, there is the splicing, one of the components of the spliceosome is heat sensitive. So I think it's forced to get degraded. So then you cannot have splicing, and all the heat shock proteins have no influence. So that's why they are expressed very quick. And also you activate the degradation system. Okay. So this is the cellular response to stress in the ER, and this is a very well studied. That is, they know from Thomas Rabbit lab, but that's what one of the things they work in the response, the cellular response to stress in the ER. Okay. And how this stress is sensed in the ER, there is a protein called BIP and a receptor called PERK. But in the basal condition, it's a monomeric and it's inactive. But once you have a heat stress, the misfolding protein will bind to BIP and will take it out from PERK. And once you have this once this PERK will oligomerize, will transduce the system, the signal that will mean that you are under ER stress. Okay. And when you are under ER stress, you want to degrade as much proteins which are unfolding as you can, and this is what we call the ERAD system, which is ER associated degradation. So there is a massive ubiquitination of proteins and degradation of proteins. So basically, when you're under stress, you tend to degrade the proteins that you cannot properly fold very quick. What happens when the quality control fails? And at any of these stages under stress, especially if you are in a continuous stress, the quality control is going to fail, and you are going to have aggregation. Aggregation usually is a failure. You know, this is really I would say was at the debate a few years ago, and I think now it is clear that aggregation is not so bad in many cases, but need to wait for the next hour for me to explain you this. But the classical idea is that aggregation is a failure of quality control, and when you have a native protein when the stress is more than the chapter of building it, and more than the degradation, if you cannot get rid of this unfolded protein or misfolded protein, either by degradation or by refolding, these proteins are going to aggregate between them and are going to generate aggregates. These aggregates are very high molecular weight and even can be visualized in the brains of people with these diseases. So basically, if you have any failure, either to degrade or to refold, you might develop this type of disease and this type of aggregation. So one characteristic of these aggregates is that the aggregates are ubiquitinated themselves. So what this means? This means they were targets for degradation, but they could not be degraded. And the common knowledge or general concept always is that once you have this aggregate, you cannot degrade it. So many times the aggregate itself, the proteasome tries to degrade them and like choke the proteasome, make it not to work, okay? Inhibit the proteasome function, and this leads to more aggregation because now you can destroy less proteins, okay? Okay, that's the concept. So we have a mechanism here by which under stress conditions we can have aggregation, and aggregation for historical reasons was associated with the neurological disease for many years, because if you take a brain of a person with prion disease, you will see disaggregate, Parkinson disaggregate, Alzheimer's disaggregate, Huntington disaggregate. Depending who found the aggregate and which type of brain, you call it different. If the Parkinson's are the Lewy aggregate, in Alzheimer's, how do you call it, intracellular aggregate. Not the tangle. Well, it has a name, I have it later, okay? So depending on which disease you have, the aggregate is a little different, but even when you see, look like a symptom of the disease. And you know, physicians gave a great service to humanity, but when they go to research, in many cases, they do the opposite. So for many, many years, because this confusion between cause and consequence, they make us believe that the aggregates are the cause of the disease, and may be that it's the consequence, and even that it is not so bad for the cells to have aggregates. So I just want to finish this, give me 2 more minutes, this first hour, saying that the aggregation is a failure of quality control is when the balance between the folding capacity is less than and then when I talk about folding capacity, I also talk about the degradation capacity of the misfolding proteins. So you have more misfolded proteins than those that you can fix, either by degrading them or just refold them. And this is exactly this. And these are the exactly over 40 human diseases are associated with formation of extracellular amyloid fibrils or intracellular intrusions with amyloid-like characteristics. So these inclusions are not soluble, so they precipitate in the cell, and this will shift the biochemical equilibrium toward the formation of bigger aggregates. Okay. Let me see. 1 more minute, okay? I show this one because I want to be exactly in the hub. So some of the common features of a protein misfolding neurodegenerative disease are that you have amyloid-like deposits in the affected neurons, and these deposits have high resistance to protease and detergents. I mean, these are really thick and very difficult to dissolve aggregates, you know. They have a very defined structure, the beta sheet structure, and they can be, and this is also facilitated detection. They can very easily be stained with Congo red or some other specific dye, and very often are reticulated. Okay. Let's take it again. 10 minutes para que vea. What do you think first?
Do you know any lab who works on cerebral palsy? That's a neurodegenerative.
How you write? So what type of what?
CP, cerebral palsy is upper neuronal. Not that.
Yeah, it's like
you are born with it, and it's
no, you mean it's genetic disease, but it's in the muscle.
yeah. Nobody knows what exactly.
They are no familiar.
No, there are a lot of cases, but nobody
Familiar,
knows.
no genetic component.
Yeah, nobody knows if it's genetic or not.
I don't know.
All right. Thank you.
Quite quick. Should we go to use the class of the other one? For the memory process. Yeah, it's application. Many of them. What's the name of this? You have one of the genetic components of the ribosome. Yeah, one of the yeah, one of the even increases the function in animal models can ameliorate the symptoms. Yeah, it's true. Actually, there is studies sometimes I don't know recent they show that beta carotenin symptoms that actually you want to integrate the beta amyloid. This is the current I think I know the no, I think that's why I'm asking. You were here when I'm asking. I said mitochondria, lysis, and yeah. I'm asking. keep the slide now. I think the slide is there. I think.
But how about not only the system like the oxidative stress in the not only you have to the market so much oxidative stress that the liver has to
but the liver also has in many of the diseases there is many cases with diabetes. That the liver, diabetes can form the liver in the pancreas. Of course, you can have a diabetes without the beta, many of the signs of diabetes can come from the liver. So, schizophrenic ataxia, in some of the Parkinson, you have a disease in the liver. It's not seems like in the brain is the most difficult thing, but many of these even aggregates in one thinking particles that have aggregates even in the cells of the gut, so people refuse to look in the brain because they get dementia, but these seem to attack just quicker the brain, but this is falling everywhere. The white cell, not the you activate the shift pathway, some pathway, but in the next one, you also increase the distress, It's like you don't take cumulative.
Like let's say, you are not sure that you are completely going to recover from that. You may partially recover.
You need to wait until the outcome. We will talk about this, exactly. Inflammation and the onset of the disease. No, you're right. Not drug will at some point they have been No, I think what people try to do is to delay the onset. You delay the onset.
But the thing is the problem is how are you going to get the early symptoms, right? I mean, once you recognize the early symptoms, then you try to do something.
No, you may want to delay the onset for everyone. Like a prevention.
I mean, once you start aggregating it, how are you going to do that? You don't get already started. So
I think they already at 40 you start aggregating in the gut. And there is people doing imaging in the brain, you can see the aggregate. Yeah, but you cannot do anything anyway.
Yeah, that's what in the gut also unless we take blood samples and but nobody is going to go for
No, but maybe look, this type of research helps you in the future because if you may have some markers, there may be some processes that are going on that you may be able to antagonize. Not who knows how, but maybe 30 years. It will be a field for the people with 20 to say.
We can read from the genomic sequence.
No, it's not necessarily.
I mean, now they have this chameleon sort of thing. They have constructed this phylogenetic like some of the machines so that.
No, but with the genetics alone, we can tell you who we have it, but you don't. We are telling why. Or use the animal model. Yeah.
Sure.
Tendency to look at all these diseases together as a group, neurodegenerative diseases, but beyond the fact that they attack the brain system, the nervous system, is there a reasonable reason to look at it?
The molecular pathways that underlie most of these diseases are the same.
But I mean, the molecular pathways are not really well understood because nobody can really say today like what is the cause of it. They have different symptoms. They attack different areas of the brain.
You can cross the mitochondria, less action, less difference of protein in the mitochondria. You have oxidative stress. You have
but these
Look, I agree with you.
Mechanisms can be a result of the disease and not the cause of the specific. Like this can't explain why Parkinson happens in substantia nigra cells and Alzheimer begins in the entorhinal cortex and then spreads in a very specific way throughout the brain, and why it's very different.
I agree with you. That's
and why aggregation, spongiform encephalopathy causes death within like a year and a half or 2 years. You get the disease, and Alzheimer's you can live 8-10 years with the disease.
No,
There's nothing in this. When you look at them the same,
of course. Yeah, like completely. I think more important than what is similar between them is what is different between them, and what makes different neural population more susceptible to one or to the other. Because all this, in many cases, is dominant. Like in mostly in the polycule, because polycule is aggregates the. But if I think this view is gone with the aggregation.
Yeah, that's like now they are trying to degrade that polycule.
Maybe
you have different polycule, different protein. Having the polycule kill different neurons, and the neuron specifically is given by the host protein, not by the polycule itself. So because all the cells, all the neurons have the same mutated. Why some neurons more susceptible? So, but
we might be missing something by looking at.
People are now I just put them all together. But this, I mean, I never heard. Most 10 years ago, people work in polycule proteins. Now you work in one, two. But the most interesting about the polycule is the specific polycule, the polyglutamine. We talk next about it. So the more interesting about the polyglutamine is the things that are different, because in different cells. Yeah, in some cases it's sequestration of the protein, so it could explain some of the particles, especially with the diseases, like even Parkinson and Alzheimer's. We don't really know what is what's in that. There are many pathways that seem to be involved, but it's not clear what is cause, what is consequence, what is the key one. It is attacking in many places, that's why we cannot.
And there are pathways that are basically involved in every disease, not just in the nervous system. That's not going to give you the answer to how to prevent or
what is the startup, it's kind of close complex, right? Like we have one disease, it's not like going to go with one disease.
Yeah, people try to understand there's something in common, but I think by that they're going to miss, like maybe finding what's different is that going to give you the answer, because what's different cannot be the consequence of all this. So now you have to say, what is this?
I mean, like, let's say, all these diseases are related to some neurons, right?
No, but even the polycule, you have the same, supposed to have the same inside.
Yeah,
and the neurons are so different, the ones which are
between the neurons
have ataxia, most cases. In most cases, you have ataxia with the cerebellum impairment, but it's not always exactly in the same place, It's not there. And you have the same toxification, so it's not clear what is but I think people, no one works
About this.
in polycule anymore, or in neurodegenerative diseases. It was 10 years ago. Now you work in a disease and you try to understand. You are interested in see what is common and what is different. Both things. Many things are done in animal models, so they have a lot of limitations.
And that it's like, we need some systemized kind of thing so that, like, you can go to all the, politics and try to make all the disease models, like, let's say, it's neuronal diseases, like, if you take Parkinson's, there are, like, some of the similar pathways which you will follow. But the thing is, cause may be different, but we'll have, like, similar kind of pathways. But the problem is, looking at one pathway, like, let's say, if you just take Parkinson's, looking at one pathway, so
yeah, but I think now people are doing the networks of disease from all the everyone dies.
Oh, at this time,
how you do immune system. I think it's in one of the organisms. It's an immune system that is adaptive. You know, you have an antigen and you create a direct thing that destroys this antigen. Here you have a thing 16 bags of cells. It's a very bad design, you know, a very high energy design, very expensive design for most of the process. You know, in the organism, it's very expensive. Not perfectly technical. I change the temperature 100 times, everyone dies. Even if I check 30 degrees. You look at all the mass extinction, the meteorite extinction. No perfect. I think you look at that's completely not taking it's a cellular distribution that the body is so perfect and so beautiful. Bacteria is much more beautiful, no? Because there is much more variation in conditions, reproduce much faster, much more efficient. The concept of beauty but for the physical man is very non-perfect, very non-perfect, completely non-perfect. What is the idea? Yeah. Okay. Can you go to look? Please look close. Okay. You see, we're going quite fast. As we finish the class today, I can even scout a little. In the next class, it's very long. Or if you collapse, I can let you go a little bit forward. So what is the relationship between the misfolding and the neuronal death? And again, here we, you know, one of the biggest, I think one of the biggest problems in, I would say medicine, but it's also in science, is to, that is not really related to this, but I wish I said it anyway. So it's the fallacy of the affirmation of the consequence, you know? You have A, then B, and then you say B, then A. And if you go, of course, to medicine, it has been, you know, if you have symptom B, always is A, you know? There is no way around. And this logically is wrong. There's no way around it. And here is the same. If you have a guy with Alzheimer's, has misfolding, then if you have misfolding, the guy has Alzheimer's, or, I mean, we define the symptoms as the proof of the disease, and then this symptom is the cause of the disease. So people look at people with Alzheimer's have this misfolding, so this is the characteristic, this is how you know they have Alzheimer's. Well, when they are alive, they have a few tests to do, you know, but in some cases you cannot distinguish between the different diseases. Someone is dead, you open the brain, you see the plaques, they're very characteristic Alzheimer's. So then Alzheimer's must be causing the disease, and as I said, this has delayed the knowledge many years, but on the other hand, we have learned a lot. We have learned a lot about protein aggregation because of this wrong hypothesis. So I don't know what in the equation whether this was a good thing or not. So you have misfolded cellular dysfunction and neurodegeneration. How these things link together? Do they link together? So what is the role of the proteasome and the UPS in neurodegeneration? I think that if it would come here, we tell me this is wrong. So I will tell you this is not completely right. This was the classical view until a few years ago, and doesn't necessarily need to be true, but I think it's a good working hypothesis to think about it. The first thing is that the aggregate, you need to think that in this original, in the middle 2000, 2003, 2004, the aggregate is evil, okay? The aggregate is what is causing the disease, okay? So people see that once you have some aggregate, the capacity to have more aggregate is incremented. One possibility is just a question of equilibrium, okay? You have something which is soluble, something which is insoluble. So obviously the insoluble, there will be an equilibrium, will get out of the solution. Second, you have an inhibition of the proteasome, which also was documented, and the idea was that the aggregate somehow choke the proteasome. So not only you are in a situation in which the balance between the production of toxic species is more than the degradation, but now the toxic species can inhibit whatever is degrading the protein. So then you have like a positive forward loop. You have like a increase and increase and increase more aggregation, and this leads in the long run to cell death. If you have independent impairment of the proteasome, for example, if you have a mutation that makes your proteasome less effective, so then you will have more aggregation and everything will go together. And the cell hypothesis, which I think is the right one with the knowledge we have now, is that the aggregates are indeed protective. So these unfolding proteins are very toxic, very dangerous for the cell because can interact with many other proteins which could be very important. But it is now, this goes a little against 1 and 2, no? This, I think the last 2 years has clearly showed that at least the big aggregates you see in these diseases are protective. Okay? You can have a lot of toxicity without big aggregates. And what is really toxic is not the big polymers, the big fibres, but the small aggregates. The aggregates of very small molecules, which are like oligomers. Okay? Are the oligomers A beta, for example? You have a question?
Yes. Is there any, are there any evidence for that if you induce proteasomal response, you can help to improve symptoms?
Yeah. There is you know, I mainly know about the fly model. There is, in the fly model, if you hit proteasome, some of the symptoms of the proteasome, you can release some of the, delay some of the symptoms and release. I would guess that something like this is in the mice, but I don't remember. And on the other hand, if you need some of the, if you have heterozygous mutations from some of the proteasome subunits, you can have the disease get worse. But again, I can mainly say about the fly. I don't know if you know about the C. elegans, and I don't know about the mouse, but I mean, I think with drugs you can inhibit and you have in cells, for example, in mammalian cells, you have the evidence, but you know, I'm not 100% sure. Okay. But in a main, this is the reason why they die. The fact that this can give you a treatment, you know, but doesn't mean that's why what is happening. Okay. So this is I just want to highlight. So protein misfolding, you guys can understand how it can contribute to these diseases, and it's a central actor. It's a central actor in the disease, and if you help the cells to fold properly, you're going to help overcome or delay the disease. This is very clear. Again, it's a fallacy to believe that because of this, that's the reason for the disease. But okay, oxidative stress is the second pathway I'm going to talk about today. So you guys know free radicals have an unpaired electron in the outer orbit, and the free radicals can cause a lot of damage also because they can pass this capacity or this high reactivity to the next molecule. Okay, so they can amplify the response. They can generate chain reactions, and this can lead to oxidation of many molecules in the cell. Okay. So there is classical example. I think Mona talked about the fact that insecticides have a very big link with Parkinson's disease. So there's many cases of chemical compounds that can increase the free radical injury. Also the inflammation and infection during childhood can have an impact even in adulthood. Okay, and she talked about microglia. Huh? Okay. So this also can generate a lot of free radicals, because when you kill bacteria, the way you kill a bacteria has generates a lot of free radicals. Also, of course, irradiation. Also, oxygen is a ridiculously toxic substance, and you know that to use this such a dangerous substance is the very we price for getting so much energy to be able to do aerobic respiration, and also age-related changes. So all these are examples of free radical injury. The classical definition of oxidative stress is the production of reactive oxygen in excess of antioxidant mechanisms, like a round definition, of course. You have oxidative stress when you have more reactive oxygen species than the one you can get rid of. And if you want to say this in nicer words, okay, we talk about altered homeostatic balance, which results from the oxidative insult. So usually when you have an oxidative insult, you're going to have a response which will keep the homeostasis. When this homeostasis cannot be kept, you are under oxidative stress condition. So I was just to mention that oxidative stress relates with the we talk about this next week. So how they relate with the protein body, but I think I will just mention when I talk about the molecule next week, okay? Because I don't have the slide here. So there is a lot of oxygen reactions that can lead to oxidative stress. This is just a cell. And what will happen if you have free radicals there? This is exactly, I guess, you guys know all this, I guess, okay? We have air, the anaerobic, there were only anaerobics at the beginning of the life, you know, and etc., etc. So oxidative radical can do damage to DNA, and this can happen in the nucleus and the mitochondria. A very important damage is in the membrane. You can have a peroxidation of lipids, which we are going to talk about. It's a very typical lesion in this type of a disease. The membrane the membrane of the mitochondria is a very big place where the ROS are generated. It's a key place, okay? And you have some mechanisms that will try to repress the oxidative stress, okay? So when you have a respiration, you have the respiratory chain. You know that one of the products of the NADH pass here. There is a change of transfer of electrons that is done in order to create a gradient of protons, which will be used at the end to generate ATP. Okay, this is the respiratory chain. Basically, I know that you guys also know that the oxygen, you have a reactive oxygen species which are involved, or which are a subproduct of this respiration. These reactive oxygen species are neutralized normally. Okay. But in some cases, not, and that's when they can, I really don't want to go into the details, okay? But basically in this situation, you're going to have damage to proteins, damage to lipids, damage to DNA. And you can imagine how this damage in this means really damage in the cell. And these are the things you see. You have lipid peroxidation, and that's exactly what you see in people with Alzheimer's. Kind of same for it. You can have protein oxidation, which is manifested because you have free carbonyl groups. You have oxidation of nucleic acids also, and you have glycoxylation. So all type of macromolecules in the cells are badly affected by oxidative stress. And the idea here is, again, neurons seem to be a little more sensitive to it, and people sustaining that the oxidative stress is the reason why we develop this type of disease. That's the argument they use. Again, you need to assume that you live under stress, okay? Because you mentioned the ROS mutant, the sub-mutant.
Okay, so they're exposed to some stress in the environment, but it's not enough to cause any physiological change. If you go to extremes, then yes, it shows.
Yeah. That would be the concept. Maybe the role of this oxidative stress mechanism has been exaggerated a little. In most models, genetic models of the disease, but I would say that indeed I remember at least two models in which the deletion of salt doesn't have any effect. I think it's FMRX and Frederick ataxia. In these cases, you have no deterioration, or not suggesting that oxidative stress may not be involved in the disease, at least in these two. In polyQ, in some of the other diseases, when you, at least, I try to remember, if you have, I will check, it is a good thing to check. When you overexpress some of these detoxification enzymes, you release some of the symptoms, at least in the fly models. And I believe for some of them in the mouse too, because that's where the paradigm was established.
For polyQ, if you overexpress salt, you get.
I think more the tau. And in the dopamine, like alpha-synuclein. Let's just not go into the biochemistry. So if you have, what happens, of course, when you have oxidation of molecules, you're going to have trouble. If you have lipid peroxidation, then you will have damage to cellular organelle membranes. Why the membranes are important? You know, what is the main problem here? The membrane will preserve gradients of protons, gradients of electrons. So they will be very, very important for the membranes which are involved in the nervous system, and also from the membranes in the mitochondria. So when you have the membranes of the mitochondria which are not working so well anymore, now you're going to have less action potential, less differential of potential, less production of ATP. It's like a vicious cycle, okay? So you also have protein cross-linking and fragmentation, and you also have, of course, DNA damage, which can result in more damage to the cell by mutation, okay? One characteristic I would say mark on the Amyotrophic disease is the presence of lipofuscin. And lipofuscin is basically lipids which result from the free radical peroxidation of the is damaged lipids which result from this peroxidation by free radicals. And usually you can see it in myocardial cells and hepatocytes, and I think I have a picture here how they look. So the oxidative stress can really affect all type of molecules and produce all type of cell lesions in the cell. Okay. So the classical view is that you have a damage, at the start of this damage, and you can use antioxidants, but at the end you always get something. Okay, that's the concept I want to give you. The idea of this class, I want to give you some background because the rest of the class, basically we have guests from different places, telling you about, which I think is very nice because we try to take from all the different aspects of a neurodegenerative disease. But I think we believe that you need like a basic ground on what is a little the history of the biochemistry of this. So I know it's not the most fascinating class today, but I think it's quite necessary to understand it. So one thing that is very important for neurons is the maintenance of the calcium homeostasis. This, the calcium is confined to certain places in the cell, okay, like the sarcoplasmic reticulum in the muscle, So it is very important to be compartmentalized, okay, because the calcium is a very important signal molecule. So again, when you lose permeability, when the membranes are damaged, you may have leakage of calcium. You may have not such a big gradient of calcium when you should have it. It's the same concept. The distribution of ions in the cells is very important. It's not just the ATP, it's not just the synapse, okay? So when you have this is just when you have increase on the calcium, you have a lot of effects which affect again all sorts of molecules. So not only you have a direct effect of the oxidative stress in the how the molecules work, but you have many downstream effects, some of them which mediated with the alterations in calcium metabolism, which are killer for the cells. Okay. So the mitochondria has a key role also in this cell disease because the mitochondria, as we said, is a place in which the energy is produced and which has this production of energy has a cost that a lot of free radicals are generated. Okay. And we are going to go now to the last pathway by which the neurons will die. So basically you could have protein misfolding to lead to the collapse of the cell. You can have oxidative stress to end up killing the cells by killing the function of the cell. And you have cases in which when the cell is going to die, it's going to do apoptosis. And again, I'm going to go very quick on the thing. We have a lot of time, so I'm going to just give an introduction to the polycubic because it's not we need to have like 90 slides next week. Okay. So cells are balanced between life and death. All of you know what apoptosis is, no? All of you what is okay. So basically, you have damage and you have physiological death signals, so this will lead you to death. And when you go to apoptosis or not, it's due to a balance between the pro-apoptotic and anti-apoptotic proteins, and in some cases, the excess of oxidative stress and damage is going to activate the pro-apoptotic proteins and inhibit the anti-apoptotic proteins. And this, I think, this is I think it's kind of like just I don't think you guys are many all of you biologists, so all of you know what apoptosis is, how important is apoptosis during development. It's not just to kill cells, but it's also to produce organs, and it's used to produce the fingers, it's used to produce adult brains. Apoptosis is very important. Apoptosis is very important to kill whatever cell recognize our own antigen. Okay. 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 So in some cases, some of the cells die by apoptosis before dying by apoptotic collapse, and I think that's it. Basically, that's what I want to tell you. Okay. Some of the death of these neurons is going to be by apoptosis. So the cell will sense that it's better to die than to keep living like this because the possibility of spreading the disease to other cells. So some of the cells are going to go by apoptosis. Let's just, any question at this point? This intends to be like a general, this intends to be just class 4 reasons. Intends to be a general introduction to the neurochemistry. And again, not all what I told you is completely updated. Okay. Okay, so I'm going to talk in the next few minutes about a certain type of disease, which really is many types of disease together, which is expansion-related disease. And next week I'm going to talk about the flying models. We're not going to have, of course, no, unless we are until 7 here. For work, okay. So I'm going to just introduce you very quickly the trinucleotide repeat disorder. So there is a historical reason for this. Many neurodegenerative diseases exhibit anticipation. So the typical case, you have a grandfather which has some ataxia, some problem when he was like 60, 70. Then you have his son, your father, which gets the disease when he's 50, and then it's you who get the disease when you are 30. So there is anticipation of the system, of the symptom over the generation. So this is has been noticed. This type of disease has been known for many, many years. This disease can be autosomal dominant. So usually they are dominant. In most cases, there are some cases of recessive disease. Okay. Main clinical disorders as well as the few. Okay. Just tell you this. There are a lot of them. Okay, that's the concept. Mutation causing this disease involves expansion of trinucleotide repeats. So basically, what happens if you, in all these diseases, a part of the genome that will carry some of these repeats, for example, CTG or CAG or CGG, or you can read them, will have maybe 10, 15 repeats. The normal individual will maybe have 10, 15 repeats of this sequence in the DNA. But the sick individual is going to have maybe 80 repeats of this triplet in the DNA. It's always this type of triplet, but not this other type of triplet. Okay? So you always have this type of expansion of a triplet. That's why it's called trinucleotide repeat disease. Usually, for most of the diseases, the normal people have less than 30 repeats. The people who are mildly sick with these diseases have around 50 repeats, and the people who are very sick, I would say like the one who maybe the grandfather got it when he's 60 or 70, maybe we have 40 or 50 repeats, but the guy who got it when he's very young, we have more than 100 repeats. There is a really growing number of trinucleotide repeats. I mean, repeats people knew disease that people didn't know what is the mechanism. Many of them have been shown to be mediated by trinucleotide expansions. So you have expansion of these repeats in the genome. Yeah.
Can you give an example of such a disease?
Huntington. Other one? Manchado Jose disease. You have I will give you a lot of them. Okay, there is a lot of them. Some of them, like Friedreich ataxia, don't manifest only in the brain. Friedreich ataxia, you have ataxia, sensory ataxia, you have cardiomyopathy and diabetes. And I believe this disease is quite interesting because we really don't understand people don't really understand what is happening, even if you know what is the gene affected. In many of these cases, we're going to see I think I have it in the next, okay, yeah, in the first slide. But in many of these diseases, the repeat, I told you, is in the DNA, okay? So this repeat is in the DNA. That's the only thing that unifies all these diseases. All of them are originated, or the cause is a repeat of this trinucleotide in the DNA, okay? So how can it be possible that the one guy has 50 repeats, the son has 70 repeats, the grandson has 100 repeats? This is because during the replication of the DNA, the DNA polymerase will go over, will count, let's say, will how do you say it in English? Slide, okay? Will slide to the DNA and will make much more repeats. secondary structure of these regions in the DNA may also be important for this sliding of the polymerase. Okay. The repeat expansion usually takes place in the germline, but there is a few cases in some animal models in which also can happen in the soma. Okay. Yeah, that's exactly. Okay, that's exactly what it is. Okay.
When people have the large number of repeats, do we see a mosaic in the person itself? So, for example, if the soma had 100 repeats, maybe in some brain cells they will have 200?
That's a good question. It's assumed that most of the expansions have, I don't think people, maybe someone checked. It's also so difficult to determine how many repeats you have, because how you sequence it,
You take flanking regions with arbitrary repeats and you just run it on a gel.
No, but the
So
PCR may not work.
We need an enzyme that is sensitive that won't screw up.
Yeah, so I remember at least most of the cases, most of the individuals are homogeneous. That's the concept. There's a few cases in which somatic expansion has been documented, but yeah, I feel silly. I don't know the right answer, because yeah.
In the replication process, are these extra repeats at the expense of other triplets, or is it just an addition and then you go back to the
in addition, yeah, because I think you can form a structure of these repeats, so the polymers go and they go over again over the same. So that's what happens.
They go and then continue.
And this is to be especially sensitive in the somatic tissue. Sorry, in the germinal tissue, more than the somatic. Maybe even a reason for that. I don't know whether the something is different. But there is at least from the animal model it was the case. But I don't know if when they take from a person, it's a good question. So the expansion is made by misidentified. Okay. It's just that's the way it happened. Okay. That's it. Okay. During the replication, this is the most accepted model. Okay. Although also this expansion could happen during DNA repair, and I think this is the somatic case. Yeah. That I don't want to go into the mechanism. Why I put so many slides? So I told you that the expansion of this repeat is on the DNA. Okay. Now you can see the expansion in many places. In some diseases, the expansion is in the promoter. Okay. If you don't expansion in the promoter, what you will get? The disease is dominant or recessive. Okay. Usually it's recessive. Most of the time it's recessive because this will lead to a decrease in expression. Usually, if you have one less copy, the system will compensate, but if you have two less copies, now most of these diseases are recessive. You have also mutations like a Fragile X, which are in the 5' UTR, which also, in most cases, are recessive, okay? Because they don't form a dominant negative. The idea when you have a dominant phenotype many times because you have a dominant negative molecule. Okay? And when you have in introns like a Friedreich ataxia and other diseases, also is recessive. So most of the cases, in many of the cases, the repeats are in non-coding regions, either the promoter, 5' UTR, 3' UTR, or introns. In these cases, at least in most of these cases, the disease is recessive. Or the strong forms of the disease are recessive. In most of these mutations, the penetrance is quite high. So usually, this means that the reduction in the expression is quite high. So the other type of disease, which is the most, known, is when the expansions are in the coding sequence of the RNA. And many of these expansions are CHE, which is encoding to glutamine. So So that's why many of these diseases are called polyglutamine diseases also. Okay. So how do the expanded repeats cause disease? When you have non-coding expansion, you have repeats in the RNA region. In some very few cases, you may have altered RNA function. So the RNA molecule itself can be toxic for the cell. And we have repeated not in DNA non-coding region intro promoters. You have reduced transcription or translation of the protein. Okay? Yes.
Are they going to affect the, if they're in the introns, they are going to affect the splicing and are they going to affect the splicing and all, like if there is a CAG repeats are in the introns?
Yeah, if you have a huge CAG repeat in the intron, many times interrupt the polyprimate tract, or you need the splicing. So you have one splice form, or you have I think in Friedreich ataxia, you have the repeat slow down the polymerase. So the polymerase just fall. Indeed, if you treat the cells with Friedreich ataxia with a substance like tricostatin A, which is going to open the chromatin, now the polymerase will go through, and you will have more expression. So I believe most of the cases not exactly splicing, but more transcriptional elongation. Because you have a huge repeat which has a very strong secondary structure.
I mean, the mRNA undergoes this nonsense mediated decay and those kind of stuff, so that avoid.
You think we'll go to an MD? Yeah, but this is happening in the nucleus, no? So even if the RNA is degraded, you will have less final product, no? You have less protein. Yeah.
More general question about what we talked about earlier. So these proteins, for example, in Huntington, they're expressed basically from fertilization, but the symptoms only appear at the 30s, 40s, 50s. Is the protein accumulated during teenage years, or do we see an abrupt increase in protein in later stages of life? Because this can distinguish the two models.
I think there is an accumulation, but the problem is the accumulation may be a benign sign.
Yes, but again, if I take a person with the Huntington gene at age 15, he's not going to get the disease until 30. Do I already see considerable aggregate?
Yeah, that's a good question. In animal models, you do. In animal models, you do. But you could say that the aggregates are part of the defense mechanism. Okay. So the question is that some experiments have never been done or never clear. Like if you take a mouse and you express the protein for 1 day in the young or for 1 day in the old, this will distinguish the two possibilities, I guess. So this has, to the best of my knowledge, there has never been such an easy experiment. Never been done.
Actually, maybe you can even do it in Siergans. There is the Eutz-Cohen model with the AB, and there's a polyQ model also. And you can
I start with Eutz. We did it. We did
each
the slide.
reproducible, yes, for 1 day and then shut it off. And what's the effect?
Yeah, the old one is more toxic.
Yes, but if you do it in the young one, does it affect it at an old age, premature symptoms?
Oh, yeah, we don't know yet. It may, but doesn't really matter, no? Because if you want to show that neurons are more sensitive, we are going to discuss this next week because I'm going to show. In the coding patch, you have repeats in protein coding sequence. So you get, in this case, the repeating coding sequence has the dominant effect. Okay. Okay. So I think we're going to stop it here, and just wanted to have an introduction, please, and next week we talk about some of the animal models to work on this disease, okay? Any questions further? No? Thank you.
Whatever the protein we show that is